Our final presentation for this workshop is from Paul Carlson. Paul is a principal investigator in the Laboratory of Mucosal Pathogens and Cellular Immunology, Division of Bacterial, Parasitic, and Allergenic Products, Office of Vaccines, Research and Review, Center for Biologics Evaluation and Research at the Food and Drug Administration. Paul is co-chair of the FDA Microbiome Working Group and the Joint Agency Microbiome Working Group. His regulatory responsibilities include product review for fecal microbiota transplantation, define live biotherapeutic products, and bacteriophage therapies. I also want to thank Paul for presenting at this late hour on the East Coast. The title of Paul's presentation is Regulatory Considerations for Microbiome-Based Therapeutics. Paul.
Paul, I'm not getting your audio through. Let's make sure your mic is correctly set.
How's that? Better?
Sounds great. You're ready to go.
All right. Double mute because I called in on the phone. So thanks to everybody, the organizers for the invite and for everyone for sticking it out for the last talk of the session. Hopefully, so I like doing these things to get the point across that the FDA is here to help. And hopefully by the time I get through my talk, you'll agree with that statement. I did see one question in the Q&A box after the last talk about the difficulty in getting these things through the FDA. And hopefully I'll make the case that that's not really an issue by the time I'm done talking here. All right, let me see if I can get this to start moving. There we go. All right, so I always have to have my disclaimer. My slides are all approved by the FDA, but they can't control what comes out of my mouth. So my comments do not bind or obligate the FDA, particularly my answers to any questions that come up at the end. So I typically structure my talks like this. I'm going to give an overview of INDs or investigational new drug applications. That's really the stage where all of these types of products are going to be. We don't have any licensed products in these classifications. And specifically today, I'm going to talk about fecal transplant and live biotherapeutic products, which some may call probiotics, but hopefully I'll make the case for people to stop using that term in this context. So again, my role at FDA, I'm a product reviewer or chemistry, manufacturing, and control reviewer. So I'm really going to focus on considerations for that aspect of these products when we get to that point. So IND, for those who don't know, is investigational new drug application. This is basically what you have to file with the FDA in order to do a clinical trial for an experimental drug. It exempts the investigational drug from pre-marketing approval requirements, so you can use it. And there are some other legal issues with shipping unapproved drugs across state lines, and it allows for that as well. But our objective in reviewing an IND, and whether it's phase 1, phase 2, or phase 3, our primary objective is always going to be to ensure the safety and the rights of the subjects in the investigation. Safety is always going to be the main concern and the main focus. In phases 2 and 3, we also want to help assure that the quality of the drug is adequate to permit quality of the evaluation of the drug is adequate to permit evaluation of its effectiveness and safety. So really looking at how the study is designed, controls, placebo groups, things like that. I think this is pretty straightforward, but if we look at the area that I'm really referring to here, we're not talking about discovery and development or preclinical models. We're talking about clinical trials, phase 1, phase 2, and phase 3. And you can see the first thing on the list for all of them, again, is safety. The main difference as you go across the phases are the number of people that are included in these studies. And this will allow for a determination of efficacy in the later phases. And the ultimate goal here being biologics licensure application and a license to market your product. So I put this slide in just to make the point that we see a lot of different sponsors of INDs. INDs are not just for clinical or for industrial sponsors. They're not just for products that are going to be marketed for licensure. These are for all studies going into humans, whether it's a clinical research study at an academic institution, or whether it's a large pharmaceutical company doing a vaccine trial or some bigger trial. So various sized companies, individual researchers, both clinical and bench, and other government agencies. These are all sponsors that we see of INDs. Again, we see commercial INDs and we see research INDs. While some requirements may be different at certain phases, all of these studies that involve using an experimental drug, or biologic in our case, in humans require that this is done under IND. So sponsor regulatory experience varies, as you can imagine from just the list above. And so we highly recommend that at the points in development where the opportunities exist, and I know people are always going to say, well, we can't meet with you as much as we want. Well, I still encourage you to meet as often as you can. The one thing I will really hammer home is the importance of a pre-IND meeting. You can have a meeting with us prior to coming in for your IND, coming in with your actual original IND submission, and I think that's a very useful thing to do. And I'll make a case for that in the next few slides. One question I get all the time is, what about CGMP? You know, this contract manufacturing company told me that I have to pay them $1 million to get my CGMP lot manufactured for phase 1 for my trial, and we don't have the money to do that, so we can't come to the FDA. Well, it's not true. CGMP is required for all manufactured products. It assures that the drug is safe, has the identity and strength, meets quality and purity characteristics that it purports to. This is straight out of the CFR. That said, we see different levels of what CGMP means based on what phase of investigation you're in. So again, here from this guidance, CGMP for phase 1 is not expected to be as extensive as for later phases or for an approved product. Can you manufacture your biologic for your drug in your research lab? Maybe. It's going to depend on a lot of things, including what else you're growing there and what controls you have in place to keep things clean. But that's not to say that it has to be done in a CGMP manufacturing facility, like some would perhaps lead you to believe. So again, meeting with the FDA is important. We highly recommend the pre-IND meeting. This meeting will provide you with the feedback you need early on in order to come in with your IND, have all of our initial questions already addressed, and that will allow you to move into your IND study without being placed on clinical hold early on. So the way this works is you, the sponsor, provides a briefing package to us with specific questions 30 days prior to the meeting. CBER then assembles a review team. That review team will consist of product reviewers such as myself, clinical review team that's going to look at your clinical study plan, statistical reviewers, toxicology reviewers in some cases, though for the most part we don't require tox studies for live biotherapeutic products or microbiome-based therapeutics, however you want to refer to them. That doesn't mean that there's no instance where we would require such a study. It really is going to be product specific. Then we'll meet, so basically you provide questions, we provide answers to those questions in writing, and then we can have a teleconference to discuss those questions further. Currently, we're not really doing those teleconferences. It's a little challenging with everyone working from home, but this is typically how we would do it. We would all be in a conference room, and if you had additional questions that we needed a response to, or that we needed to discuss more amongst ourselves, we could do that by putting the sponsor on mute and having further discussion. Again, we can touch on, this meeting can touch on CMC, preclinical, and clinical topics. What you should provide in this meeting packet is rationale for your use of your product. What are you doing? What's the objective of the study? A description of your product. I'll get into this a little bit more in terms of the CMC for LBPs in a little while. What's your indication? What are you treating? A draft of your, or summary of your protocol, whatever you have. The more you can provide in this application for your pre-IND, the more information you're going to get back and the more useful it's going to be. And again, any specific questions you have for us, and I don't mean to say that your question should be, is all of this okay? But specific things that you would like to know about your manufacturing or your clinical trial proposal or those things. And then we discuss that and we provide that feedback, and then when you come in with your actual IND application, hopefully all of the issues have already been addressed. and you can fairly quickly, we have 30 days to review the original IND submission. And at that point, you should be able to move right into your study, assuming everything has been addressed from the pre-IND meeting. All right, so now I'm going to go into some specifics. I'm briefly going to touch on fecal transplant, and then I'm going to spend most of the time available on live biotherapeutics. So just a brief history of FMT at FDA. Prior to my arrival, the FDA held a workshop in May of 2013 in collaboration with FDA. I'm sure some people on this call were at that workshop. It was well attended by all sectors and all interested parties. At this meeting, the FDA noted that use of FMT in clinical studies to evaluate safety and effectiveness are subject to FDA regulation. You'll hear some people say that the FDA decided at this point that FMT was a biologic and subject to regulation. That's not true. It's been that way from day one. It is a biologic. It's just that at this point, it was made very clear to those using it that this was the case. A couple months later, we issued a final guidance allowing the use of FMT for the treatment of C. diff not responding to standard therapies under enforcement discretion. This doesn't mean that you can do whatever you want. It does mean that you don't have to have an IND in order to treat someone with FMT for C. diff. If we do find problems, we can enforce the IND requirements at our discretion under this guidance. Since then, we've issued 2 additional draft guidances. Neither of these has been finalized. You can see my slide still says comments under consideration for the 2016 guidance, and I have no other updates to provide for that. I know that people on either side of the argument have very strong feelings about whether that guidance should be finalized or not. That's above my pay grade, and I can't really tell you anything else about that at this time. So let's just talk about some of the challenges in regulating fecal transplant. You can imagine this is a complex product. Product consistency can be a challenge when you have different donors and your donors change over time. So how do we control some of these things? And again, our key concern is safety. How do we ensure safety? And we do that by screening our donors and making sure that that the protocol for collection of donations and the donors are safe, or at least as safe as we can determine based on the tests and the procedures that we have. So questions like how often should donors be tested? Should off-site donations be allowed? I always argue no, they should not, because especially if the donors are being paid, there's financial incentive for lying. And so I believe personally that all FMT donations should be done on site. Testing of donor stool, what organisms should we test for? How good are our tests? I'm going to talk about this a little bit. I was going to say, typically when I give these talks, I say we have a really good handle on that, but that's changed recently, and we'll talk a little bit about that. There have been a lot of discussions about perhaps the more long-term effects of manipulation of the microbiome here, and the fact that the microbiome may be involved in a lot of different diseases and conditions. And so changing the microbiome may have long-term effects. These are hard to really get a handle on when you're talking about shorter-term studies. And then on the other side here are just questions about how we characterize the effectiveness and the consistency of a product. How do we determine potency of a stool product? Are there specific organisms that we need to look for, or specific groups of organisms, or functions provided by those organisms? And the science isn't there yet for any condition or any indication that FMT is being used for, but hopefully we'll get to that point in the future. And we'll be able to answer that last question about what is a good potency assay, because currently it's really just a measure of how many organisms you have present in your product. But let me talk a little bit more about recent developments in donor testing. I've heard a few people mention recent FDA safety alerts surrounding FMT. This one's from last year, a couple of years ago now, about multi-drug resistant organisms. This was following a transmission of ESBL E. coli in a fecal transplant. More recently, we released this safety alert regarding transmissions of enteropathogenic E. coli and Shiga toxin producing E. coli in fecal transplants. And then finally, the one that I think I've heard discussed a few times here, and this isn't to say like the other two that we've seen transmission. We have not seen transmission, but the fact that SARS-CoV-2 is showing up in stool leads us to feel the need to warn the public that this is a possibility. And we're still looking into this, and I don't think we know for sure that this virus can be transmitted by stool. And my research lab at FDA is actually looking into this now, and I'm sure others around the country are doing the same. So recently, I published a paper in Cell Host and Microbe that had a list of donor screening recommendations, which you can see here. I'm not going to go through it all, but these were basically an overview of the things we see people testing for their stool donors. And the timing of testing is dependent sometimes on the use. Sometimes it can be bookend testing. Sometimes we want sponsors testing every donation that comes in. But you can see in red here things that we've added. We've always required testing for Shigella toxin, or Shigatoxin, or Shigella, or Shigatoxin producing E. coli. But we've changed that now after the safety alert to require that this be done by a nucleic acid amplification method. Typically, this is PCR. EIA seems to be missing some cases. We did not include EPEC testing in our requirements due to what we thought was a lower prevalence of this organism, but after seeing multiple transmissions, we feel that it's important to include EPEC in our testing regimens now. And then we do now require some form of SARS-CoV-2 screening. What that means right now, without a defined or a validated stool assay on the market, could be a combination of symptom tracking and also nasopharyngeal testing, or other types of testing that can be done on these donors periodically. But we need to have some indication that these donors are not shedding this virus. And at this time, we have put all INDs on partial hold. It's a partial hold because we're preventing the use of any stool that was generated after, product generated from stool donated after December 1st of last year. And this is just the paper in Cell Host and Microbe that I mentioned earlier. All right, I see I'm short on time, so I'm going to speed up a little bit and get into LBPs. LBPs, a live biotherapeutic product. Again, this is what we're referring to, a bacterial product or any product containing live organisms that is being used for the prevention, treatment, or cure of a disease or condition of human beings and is not a vaccine. We're excluding what you would classically call probiotics. Those are supplements. But if a probiotic that's marketed as a supplement is used to treat a disease or condition of human beings, it is an LBP, and you are required to have an IND to study that condition. So CMC for LBPs, we need sufficient information to assure the proper identification, quality, purity, strength of the product. And this is going to change over time, and we're going to require more information over time, and that's submitted as amendments to the IND. So some things that you can include or you should expect to be asked for would be, what is your strain? What's the name of your strain? Where did it come from? What's its passage history? Relevant genotype and phenotype information, and ideally a full genome sequence. I can't say this is going to be required for everything, but with how cheap sequencing is now, I don't see a reason why you wouldn't want to have a full genome sequence on your organism. We need to know antibiotic resistance profiles for clinically relevant antibiotics. What are you going to use to treat individuals if there is a breakthrough of your product in your patient population? Information on cell banking, drug substance and product manufacturing. How are you storing your bugs? How are you making these products? Stability data for the duration of the treatment phase of the study. So you don't need 5 years of stability data to start out. If your study is 1 month, you need a month of stability data. Under manufacturing controls and release testing, you're going to need some sort of potency measurement. Typically right now, that's a measurement of viable CFU for the product. But if you have any additional properties that can be indicative of potency, then that'd be a useful thing to add to the test. Finally, bioburden testing. This is the demonstration of the absence of undesirable organisms, and this is done under USP 61 and 62. You can Google those to find it. One looks for total anaerobic or total aerobic microbial counts. We may also ask for total anaerobic microbial counts depending on your manufacturing processes. And USP 62 looks at specific organisms of concern. And additional testing might be required depending on your population, your patient population, or if there are other organisms of concern manipulated at the same facility. We updated our guidance in 2016 to provide a little more flexibility for those using commercial products that would now fit the definition of an LVP. While these are generally considered safe in healthy adults, there are potential safety issues depending on the patient population that they're being used in. And so what we allow for now is you can request a waiver of the CMC information. Sometimes it's hard to get the CMC information if you're not the manufacturer. And the company that makes the probiotic doesn't necessarily have the incentive to provide you that information because they're already selling that product. So you can, again, request a waiver. If the waiver is granted, I'll tell you on the next slide how that waiver is granted. But if it is granted, then the label of the commercial product is all you need to satisfy the CMC requirements. If it's not granted, then you need to submit a full CMC package. In cases where the IND sponsor is not the manufacturer and the manufacturer does not want the IND sponsors to see all of that information, we do have a mechanism called the master file mechanism, where the manufacturer can submit all of their information to a master file that's then cross-referenced by an IND. The IND holder never sees it, but it gives the FDA all of the information they need to fully review the manufacturing and the IND. So these are the criteria that have to be met in order to be granted the waiver. The LVP proposed for investigational use is lawfully marketed as a conventional food or dietary supplement. This is something that you can go to a store and buy. The investigation does not involve a route of administration, dose, patient population, or other factor that increases the risk associated with its use. If it's a capsule, it needs to be taken orally based on whatever dose is recommended on the label. And if the patient population is immunocompromised or if there are some other concerns, then we may ask for more information. The investigation being performed cannot be intended to support a marketing application. If you're trying to market an LVP for a drug indication, you're going to need to provide the full CMC package. And the investigation is also conducted otherwise in compliance with all requirements for the FDA. And again, you submit this request, and if we grant it, then all we need is the label of the product to review the CMC information for your product. We've also written a review. This is, I published originally as a book chapter, but it's also as a review in Microbiology Spectrum on the use of or regulatory considerations for these types of products. It pretty much goes through the things that I've talked about today. And then we also have the guidances that I was showing excerpts from, and I have links to those coming up and throughout the talk. And I can make the slides available to everyone. So just final thoughts. I think we all know that interest in these types of products is increasing and continues to increase. We feel that our regulatory approach is science-based and has allowed these novel approaches to be tested safely in the clinic. And we'll continue to do this, and our decisions are always going to be science-based. And here's some additional resources, guidance documents that I mentioned throughout my talk, some contact information and other websites that may be of use. And you can also, anyone who has any questions can just email me. Our email addresses at FDA are pretty simple. It's just paul.carlson@fda.hhs.gov. And I think some other speakers here can tell you that we're pretty open to discussions about these types of things, and I'm here to help anyone who needs it. And with that, I can take any questions.
Thank you very much, Paul. And that first question that you answered, and maybe we'll come back to that was asked to Stephanie. I actually purposely withheld that. Sorry to the attendee, but I knew I was going to ask the question from you. It was around the ease of approval of microbiome-based therapeutics. But I think you've mentioned that, or at least highlighted the fact that not only is the FDA open to these discussions, but that you're willing to work with investigators and obviously considering safety, think about lowering the barriers for approval, but maybe you can comment just on how do you, I don't know how you quantify ease of an approval.
Yeah, I don't. So I think that there's just this thought in the field as a whole that the FDA is really going to be an impediment, and we're not. And I think you can vouch for us here. I think there are a few things that people think they have to do that they don't actually have to do. One being the CGMP lots and the really expensive manufacturing for phase 1. We want to make it easier to get into phase 1 to try these things out and see if they're going to if they have promise in humans. We also, for this product class, typically don't require toxicology studies, like I said, because these are human commensals. Why do I need to see what happens when you put them in a mouse? And if there's no signal, does that tell me anything? So we really don't want you to have to do studies that aren't going to inform our decision making. And so we've been really flexible there. The other big thing is typically we don't expect to see trials in healthy populations, just to take your drug and go right into a healthy population. You can go into phase 1 in your indicated population. And I think that's an important step for people to understand. And really that can help speed up the process overall. We do also have various programs like Fast Track, Orphan Drug, and various other incentive programs that can speed up the review. You may have heard, I think I saw a headline today about some COVID vaccines that got Fast Track designation, things like rolling BLA review and additional meetings with the FDA that can kind of help with those things. But at the end of the day, what you need to get your drug approved is evidence of a safe and effective drug. And that's what it's going to come down to. The trials have to happen, and placebo-controlled trials have to happen in order to get it approved. So it's science and perhaps some of the investment in getting those trials done that I think are the most important things right now.
You actually unknowingly answered the next question about considerations of going into HMVs or healthy normal volunteers versus disease populations. But we have more questions. So from our attendees, for donor screening, do you have to have some sort of a diversity index which seems to be correlated with healthier treatment respondent individuals?
No, because, and typically we don't ask for any sort of microbiome data at all, because right now the science doesn't tell us anything in terms of what we can do with that data, right? Diversity seems like it's better, but is it better just because you have a better chance of getting the organisms that matter with a more diverse sample, and a less diverse sample that just happens to have those might be good too. So without that basic understanding of how to interpret the data, we don't typically ask for that type of data, because again, we're asking for things that we don't know. It can't really help inform our regulatory decisions, so we typically don't ask for that.
Yeah, I assume the motivation for the question was that diversity tends to correlate with health, and so that's the sort of
and health of the donor.
Thinking about diversity as a factor.
Yeah, and health of the donor. But what does that mean for the recipient? How does that interact with the bugs that might already be in the recipient and the resulting diversity that you end up with in that individual? It's just hard to draw a line there. I think it's important for sponsors to think about that in terms of showing efficacy. If they think there's a specific signal that's important, and they can build that into a potency assay or something like that to have what they believe is going to be a more effective drug going into their trial.
And I've heard people like Michael Fischbach and others argue about this diversity point, even though there's a correlation, that there's many reactions and chemistries that happen in a diverse microbiome, some of which may not be good for you, right? And a large fraction of which we just don't understand yet. So, so I do agree that a rational approach makes more sense than just saying diversity is good. This next question, actually something you and I talked about. Horizontal gene transfer can move antibiotic resistance and pathogenicity factors between bacteria. Will there be guidelines regarding the permissibility of LBPs to acquire these factors?
We have not gone that route. We have bigger concerns about LBPs passing things on, perhaps, and that's why we want to look at antibiotic resistance determinants and perhaps whether they appear to be on transmissible elements. But there's no evidence of these things picking up these specific types of things that we might be concerned about. The one thing that I always think about in this context is the use of non-toxigenic C. diff to treat toxigenic C. diff infections. And yes, there is evidence of transfer of toxin genes from toxigenic strains to non-tox strains, but the level of that transfer is so low that these things should still be outcompeted by the higher numbers of non-tox strains that are in there. So there may be some concerns for certain organisms that might be highly adept at picking up DNA. Enterococcus comes to mind. But in general, no, this isn't a major concern right now.
Thank you, Paul. And then there's one more question. I think it's quite an important one. So even though we're a bit over, I'll ask, has any genetically modified live bacteria been approved as a drug? What is the biggest hurdle or challenge in passing regulation for a GMO?
So no live biotherapeutic product, no bacterial product in this context has been approved, period, GMO or not. But we do have examples of attenuated vaccines that are genetically modified and have been approved. And these are bacterial vaccines. And this is really why, I'm in the Office of Vaccines and we're the ones regulating these because we have the experience with this type of product. The biggest hurdle or challenge in passing regulation is your clinical trials. Show the data, show safety, show efficacy. Depending on your genetic modification, the requirements for preliminary data for animal models for toxicology might be different. But it really comes down to what that modification is and if that modification leads to any increased concern about safety. I can say that we have genetically modified organisms in clinical trials. I can't really say much more than that. But in general, I don't see that there's a bigger bar for these types of organisms to pass. You have to show safety and efficacy of your product.
Perfect. Thank you very much, Paul. That was very
no problem.
Informative. And before I turn it over to Steve to close the workshop, I'd like to just thank folks from me as well. First and foremost to the speakers for really a wonderful day of science and thinking about where we are with the microbiome and where the field is headed. This webinar, the workshop would not have been possible without Leslie Maxfield and her team, Rachel Dempsey and Lauren Breyer, who again worked tirelessly behind the scenes, and Dorit for her inspiration and her help along the way. Personally, I'd like to also thank Henry Schreiber, Reem Abdel Haq, Jessica Griffiths, and Joe Blocker. These are members of our laboratory who helped screen some of the questions that Steve and I asked. And finally, to the attendees for participating in the workshop. And also thank you to Steve.
Thank you, Sarkis. Yes, I want to echo the thanks to everyone. This has been really a spectacular day. And I don't really have much to add in terms of closing, but I did want to remind everyone that the videos will be available online at the Trish website probably sometime in the next couple of days. And also there is a plan for a meeting report publication that should be submitted to a journal, presumably within the next three to 4 weeks. So look for that, look for the videos, and thanks again for participating. The meeting now is closed, and hopefully everyone will have a great upcoming week. Thank you.